Analysis of chromosomes can tell us a lot about an individual. Typically, you would use a karyotype or a photograph of the actual chromosomes to sort of get the counts and try to see what was different about chromosomes. What we see is that sometimes they're not passed on correctly. Sort of the general category of this is a mutation. A mutation is the alteration of an organism's DNA. It could range from a change in a single base pair to the insertion or deletion of whole segments of DNA. It can result as a process or a malfunction of the process of mitosis or meiosis, exposure to a physical or chemical agent like a mutagen. But what we see overall when we're looking at chromosomes is that if a mutation affects a group of genes or an entire chromosome, we call it a chromosomal mutation or a chromosomal abnormality. That could be something where meiosis fails and the chromosomes don't separate properly, which is nondisjunction, or it could be something where segments of DNA are exchanged or broken or removed. And we'll see various varieties of what this can do to you throughout this video. Nondisjunction is probably the most common type of chromosomal abnormality, and what happens here is that the chromosomes don't separate properly. Traditionally, you should have 2 copies of every chromosome. You should be a diploid organism, but if you have the wrong number, you have what's called an aneuploidy mutation. Most of the time, this is going to upset the biochemical balance so much that the embryo will abort, and it leads to miscarriage. But sometimes it's going to affect it just enough to have problems, but not enough to kill the embryo. So if you have a nondisjunction mutation or an aneuploidy mutation with a characteristic set of symptoms, we call that a syndrome. One of the most common syndromes that you might be aware of is Down syndrome. What I mean by not separating properly in meiosis, now this could happen in meiosis 1. Here I can see a nondisjunction mutation in meiosis 1, or it can happen in meiosis 2. Meiosis 1 nondisjunction mutations are going to hurt you more because that means that all of the gametes will be wrong. But a meiosis 2 nondisjunction, half my gametes will be correct and half will be incorrect. But it just means that for some reason, the proteins that hold the tetrad together don't separate properly if it's in meiosis 1, or the ones that hold the sister chromatids together don't separate properly in meiosis 2. There are many notable aneuploidy disorders, but you do need to be aware there are 2 major classifications. You have the autosomal disorders, which are your non-sex chromosome disorders, and your allosomal or gametic ones, which are your X and your Y chromosome. The most common autosomal disorder is trisomy of 21 or Down syndrome. There tends to be a characteristic facial appearance, weak muscle tone, intellectual disability. About half of all affected children have heart defects or digestive abnormalities, so on. The next most common is Edwards syndrome, which occurs in approximately 1 in every 6,000 to 8,000 births. These babies tend to have slow growth before birth, a low birth weight, small jaws, small mouth, abnormally shaped head. When clench the fist, the fingers tend to overlap, and fairly high number of life-threatening medical problems tends to lead these children to die within their first month. The third most common is Patao Syndrome. Patao Syndrome is trisomy of 13, about 1 in every 10,000 births, associated with severe intellectual disability, heart defects, brain or spinal cord abnormalities, small or poorly developed eyes, extra fingers or toes, cleft lip is fairly common. Now, when you look at the allosomal or gametic aneuploidies, you need to look at karyotypes to really see this. There are certain physical conditions that are common, but it's easiest to see it in a karyotype. Having more than 2 copies of the X chromosome is something we call trisomy X or triple X syndrome. It's characterized, like we said, by an extra X. About 1 in every 1,000 newborn girls have this. Although females with the condition may be taller than average, there tends to be very little change in physical features, normal sexual development, able to conceive children. There does tend to be an increased risk of learning disabilities and delayed speech and language skills. Klinefelter syndrome is XXY. This will be a male that will be infertile. it's fairly underdiagnosed because the mild signs and symptoms are common here. They don't produce as much testosterone, delayed puberty, reduced facial hair, body hair, breast enlargement. The main problem here is going to be infertility. Turner syndrome or monosomy X will be females. They tend to be shorter. They tend to have sort of a puffiness to their bodies. Webbed skin forms along the neck. About 1 in every 2,500 girls. The most common physical ones are very short stature and a female infertility. Jacob syndrome or XYY has having a male with an extra Y chromosome, about 1 in 1,000 newborn boys. They tend to be taller than average, but with really no major changes in physical features. A slight risk of learning disabilities, delayed development of speech and language skills, behavioral and emotional difficulties. And this also tends to be undiagnosed because it gets confused with an autistic spectrum disorder. Outside of nondisjunction, you can have chromosomal aberrations or mutations. Deletions are going to remove segments. Duplications are going to copy segments. Inversions are going to flip them around so they're in the reversed order. Translocations will move a segment from one chromosome to another one that's non-homologous. So translocations is like crossing over, but to a non-homologous chromosome. Each different type has various diseases associated with it. Huntington's disease, for example, is a duplication that leads to the death of nerve cells because of an accumulation of an enzyme. Philadelphia syndrome is a translocation that can cause leukemia. Prenatal diagnosis is a way for doctors to determine before birth whether a fetus has a problem. During amniocentesis, a sample of amniotic fluid, which is the fluid around the baby, is removed and sent to a laboratory for evaluation. Basically, a thin needle is pushed through the abdomen and withdrawing that small amount of fluid. The body will make more fluid to replace the fluid that is taken out, and the baby will not be harmed. Some women feel a mild cramping, and the doctor tends to tell the females to rest for the rest of the day. Chorionic Villus sampling, or CVS, is performed by removing a small sample of the placenta, which is the nourishment for the baby from the uterus. It's removed either by a catheter or a needle. In both cases, the fluid or material extracted is going to be centrifuged, the chromosomes will be extracted, and you're going to look at a karyotype to determine what is going on for the baby. When these can happen vary, but you do need to be aware that they are potentially harmful. There is a small percentage of birth miscarriages that results after this. Amniocentesis or CVS may be considered in situations in which the parents are at increased risk of having an infant with a genetic disorder. The test may be useful if you're 35 years of age or older by the time your baby is due. What we see is that overall, the older a woman is, the more likely they are to have a child with a chromosomal abnormality, such as Down syndrome. So the tests tend to be fairly helpful if you are at risk, if a partner carries a known genetic disorder or at least is related to someone that does have one.